Hello, ACA class at Patrick Henry. This is Miss Sheely with your Chapter 1 Study of Life video lecture notes. So the first thing that we're talking about this first section is called the science of biology, and there's some basic stuff that you would know, things that a basic entry-level biology student would know, but also that a freshman, sophomore level biology student would know. So a little bit of this is review, and then we'll get into some of the advanced objectives. So the two basic objectives we have: being able to identify the characteristics of science, which we went over a little bit, and being able to summarize the steps of the scientific method, which we did also go over the first day, but I just want to review a little bit more. So, of course, what is biology? So this is biology. Ology is the study of, bio is life. So the study of living organisms, but not just living organisms, it's their interactions with one another and their environments. So in biology, we're looking at all living organisms. So we're looking at the microscopic cellular level, which is the level that I prefer, and going up to the whole ecosystem, the biosphere, the whole planet, if you will. So from the smallest bacteria, single-celled organism, all the way up to our Earth and biosphere as a whole. So biology is a science, and the science comes from the Latin word scientia, which is knowledge, and knowledge that covers the general truths or the operations of laws. And we talked a little bit about the difference between a law and a theory. But these laws, this science, this knowledge, especially when it's acquired and tested by the scientific method. So the scientific method is very, very integrally important in science. So what is the scientific method? It is just a method of research. There aren't really like defined steps or sequences that you would go through. It's really a maze of a process, and we'll get to that in a little bit. But it does have some specific steps that you would go through, and you're going to have your experiment, and you're going to include observations as you go. And the key feature is that we're developing a hypothesis, a testable prediction, and then we're repeating our experiments to test that hypothesis. And this scientific method is actually credited to Sir Francis Bacon in the mid-1500s to early 1600s is when he lived. So the late 1500s is when he developed this or coined this idea of the scientific method. So remember that a hypothesis, this is just a suggested explanation for an event that you can test. So it's not really necessarily a prediction, but it's an explanation instead, is a better way of putting it. And then a theory is when we have a hypothesis that we can verify. We've tested and confirmed this hypothesis over and over again, this explanation with observations or phenomena. And so far, there's not been any evidence to falsify it. But it's not a law. It's not a fact that can be replicated over and over again. There are some disciplines, some science disciplines that don't really apply the scientific method because repeating experiments is really difficult in these fields. So archaeology, think about that. We can't repeat experiments on bones that we find, or even archaeology is not just bones, it's also just looking at artifacts. So we have a limited supply of those, so we can't repeat experiments on them. Psychology and geology, again, we're really limited, but we can still have hypotheses, and we can still support these hypotheses with observation and data. We just may not be able to replicate those experiments over time. So, of course, the traditional idea of the scientific method, we start with an observation. That observation is often a problem that we want to solve, but it doesn't have to be. And that observation then leads to a question. So we'll be focusing on that question this year. It's something that I find that students have limited experience with. So the research question. And to solve that problem, we come up with a hypothesis. The hypothesis has typically been written as an if-then statement, but it does not have to be written in an if-then statement. But of course, our good rule of thumb that we teach our students is if the IV, then the DV. But it doesn't have to be that way. It could be that the instead of saying if the bean plants are grown in light, then they will bend towards the light. It could just be that the bean plants are grown, it will bend towards the light as they grow. Our hypothesis, in order to be valid, it has to be testable. So it should also be what's called falsifiable, and that means that the experimental results can disprove it. So we talked briefly the first day about how important it is that the hypothesis is not right or wrong. Instead, our experiment supports or does not support the hypothesis. So we want our experiment to be, our hypothesis to be falsifiable. So science doesn't prove anything. Instead, we don't support it, or we support it. So this step, giving an openness to disproving the ideas, that's really what is at the root of distinguishing a science discipline from a non-science discipline. A non-science discipline is not open to disproving ideas. And then to test our hypothesis, of course, we'll have an experiment. Our experiment will have several variables, and it will also have controls. Remember, a variable is what you change. The variable that you change is the independent variable. That's what you manipulate. And the variable that you measure, or the responding variable, is the dependent variable. And then to have a really good experiment, we want to have a control group. And this is a group that is not given the manipulation or the independent variable that is part of the hypothesis. So that way you can have some comparison of what would have happened without that outside factor. All right, so just to review, here's a good flow chart, and this is a really good visual as to what would happen. So you can see it's very linear, but then once you get to the analyze the results, was it supported or was it not supported? Not only do you report the results, but you may go, if it's not supported, you may go all the way back to that hypothesis again and try again and repeat, especially if you're in a laboratory setting or clinical setting. And then here I have the ordered number of the scientific method, and then an example of possibility with it. The scientific method, it isn't as traditional. So you start with an observation. The observation is maybe there's something wrong with my electrical outlet. Your question that you'll come up with is there something wrong with my electrical outlet? Then you come up with a hypothesis. If something is wrong with the outlet, then the coffee maker will not work when it's plugged in. Your prediction, your toaster doesn't toast your bread. So you plug the coffee maker into the outlet, that's your experiment, and the coffee maker works. So now you go back and you have a new question. Why doesn't my toaster work? It's not that something, you've now had an experiment where you found out that nothing's wrong with your electrical outlet, but now you know that something's wrong with your toaster. All right, so here are the advanced objectives. So we also want to identify the shared characteristics of the natural sciences. We want to compare inductive reasoning with deductive reasoning, and we want to describe the goals of basic science and applied science. So biology is what we call a natural science. A natural science is a field of science that's related to the physical world and the phenomena and processes around it. And it's typically divided into life sciences and physical sciences. And life sciences would include anything that involves living things. So, of course, biology, but also anatomy would be in there, or anything that's ecology is also a life science. A physical science is the study of non-living matter, and that would include astronomy, geology, physics, and chemistry would be part of your physical sciences. And then we also have what's called interdisciplinary sciences. So these are disciplines that build on both the life and the physicals. And so these are like kind of combination disciplines. So biophysics or biochemistry. Biochemistry is something that I have found to be super, super interesting. Hard sciences, this is just really another name for natural sciences, because these natural sciences are going to rely on what we call quantitative data, or usually hard data or numbers. But social sciences, this is going to be a contrast, as these rely on typically qualitative data or descriptions. So this would be sociology and human behavior. You're not going to have quantitative data in those sciences. All right, so science's goal, the goal of science is just to know. Just to know. So how does the world work? We want to know how the world works. So curiosity and inquiry are really, really the driving forces behind a science. And there's two types of logical thinking. There's inductive reasoning and deductive reasoning. Inductive is going to use related observations to arrive at a general conclusion, but deductive reasoning is going to use a principle or a law, and it's going to forecast a specific set of results. And both of these are really related to the two main pathways of scientific study. So you have descriptive or discovery pathway to scientific study, or you have the hypothesis-based science. So the descriptive or discovery is inductive. You're really aiming to observe, explore, discover, maybe without a path of mind. And the hypothesis-based, this is we're going to use deductiveness and deductive reasoning. And we're going to begin with a specific question or a problem, and then we will find a potential answer or solution that you can test. So either of those pathways are fine. The descriptive discovery is also referred to as kind of a serendipity science, where you found something out by accident. And we'll talk about some places in history where science and biology has been serendipitous. All right, so scientific reasoning review. So I want you to pause at this section. I want you to read each of these 4 scenarios and decide if each of these is an example of inductive or deductive reasoning. And then I will give you, if you want to check your answers in class, we can. So at this point, I'm going to continue on, but you stop and pause. All right, so another thing that we need to look at that's more higher level than we typically teach in the high school is this idea of a basic versus applied science. So is it valuable to pursue science for the sake of simply gaining knowledge? Or does the scientific knowledge only have worth if we can apply it to solving a specific problem or really bettering our lives? So basic science or pure science seeks to expand the knowledge regardless of the short-term application. So the immediate goal of basic science is knowledge for knowledge's sake, and we're not going to think about any particular reason. We just want to know the answer, and it's not necessarily going to solve a problem. Applied science is kind of referred to as technology, but you have to be careful how you use that because it's not meeting every definition of the word technology. And this is using science to answer or solve a real-world problem. And so the problem is usually defined or even given to the researcher. It's not something that the researcher comes up with on their own. And so scientists usually carefully research and plan their research in both basic and applied scientists. Again, as I mentioned before, some discoveries are made by what's called serendipity, which is a fortunate accident or even a lucky surprise in which we are able to gain some knowledge and learn something new about how the way the world works. So what do we do after we do our science, and how do we get the word out there? So before announcing a discovery in a scientific paper, we have to submit it to the paper, and it will undergo peer review. We talked about peer review a little bit the first day. So peers are, of course, qualified individuals. They're often experts in the same research area, and they're going to judge whether or not my work or the scientist's work that is submitting it is suitable for publication. And this is really going to help to ensure that the research is original, that it's significant, that it's logical, and that it's thorough. It's presented in a way that can be repeatable, not that your procedures need to be given out explicitly, but so that other people can research a similar area or a similar way. And this is actually the exact same process or very similar as to how we request funding for a laboratory or for the scientists themselves. And so that's typically done through grant proposals. And so they also have peer review for the proposal. Scientific writing is so different than creative writing or writing for history or English. It has to be very concise, has to be accurate, but it needs to be detailed enough to allow peers to reproduce the experiments. And this is how we will learn to write throughout the year in class. It is not something that is easy typically for most students. It's very fact-based and very concise. So here's just a visual of the peer review process. So you're starting with your idea, your theory, your hypothesis, then you do your research, you write your paper, you have a manuscript, and you send it to the journal. And then it's reviewed by 3 to 5 people in a timely fashion, and then a decision is made whether they will accept it, whether they're going to send it back to you to revise it, or whether they reject it in general. And of course, if they accept it, then it will be published. And then if it's super exciting, you can also maybe have a press release where you announce it to the world. So a scientific paper contains several sections, and a good format or acronym for you to remember this is called I'm RAD. And this will include several things that we'll talk about in a second. Well, the first one, the I, is the introduction. The M is the methods and materials, or materials and methods. The R is the results. So we need to make a word, so we'll throw the A in there. It doesn't mean anything. That's why it's lowercase. And the D is the discussion. Sometimes a scientific paper will also include an acknowledgement section, or reference sections, or even an abstract, which is just a concise summary. So your introduction, brief, broad background information about what's generally known in the field, giving your rationale, like why is this important, how did you get to that area, that idea, justifying your work, and we'll also mention the hypothesis or the research question. This refers to the published scientific work of others and includes in-text citations or citations in general. The materials and methods is what you're probably more comfortable with. This is a complete and accurate description of the substances, the methods, the techniques used to gather the data. It also often includes visual diagrams or pictures, and it's going to allow another researcher to be able to repeat your experiment, and will include information on how precisely the measurements were made and the types of analysis that were used to examine the raw data. So it's not just the methods of collecting the data, but then what did you do, the methods to analyze the data as well. So the results are going to narrate the findings, but they don't have any interpretation. So you're just you're not explaining, you're just talking about or describing the results, the tables, the graphs, the analysis of the raw data. The discussion is when you get into the interpretation. What do your results mean? How are those variables related? And you'll explain any observations that you made as well. So this is really where you put the context of the published material that you put in the introduction with the analysis of your results as well, and explain why. So you're really marrying those two sections. We will be reading scientific papers throughout the year, and I will teach you how to read them, but you do want to familiarize yourself with the basic format. You could think back to your 9th and 10th grade years when you did research projects. This is very similar to a science paper format. In addition, when you do a formal lab report, your formal lab reports will contain these elements. So this is a good visual, quick reference as well. All right, so that's the first section of Chapter 1. Chapter 2 is talking about the themes and concepts of biology. So again, I'll go over the basic objectives, identifying and describing properties of life, describing how the levels of organization among different living things. So you have properties of life, and there are typically about 9 different properties of life. Some textbooks will say 7, some will say 9, but generally they're pretty much the same. So all living things are going to have an order to them. They're going to be able to respond to the environment. They're going to be able to reproduce. They're going to adapt and change. They're going to grow and develop to totally different things. They're going to have some type of internal, some type of regulation. They're going to have homeostasis. They're going to be able to process energy, and over time, as a species, they will evolve. So this is where we really ask the question, is it alive or is it not alive? And to be alive, we typically say that a living thing would need to be able to meet all of these properties of life. All right, so let's talk about order first. So organisms are very highly organized. So we have coordinated structures that could be just one cell, because remember, a living thing could be one cell, or we could be multiple cells. But even within that one cell, we have order. So our order is that atoms make up molecules. Molecules then make up organelles. Organelles then form cells, and therefore so on and so on. So you have this very similar order no matter if you're stopping at a cell, at a single-celled organism, or you're going all the way up to a multicellular organism, or even looking at the whole biosphere together. All right, so organisms will respond to their external environment or to stimuli. So they can respond to that stimuli either positively or negatively. That stimulus is going to be some type of change in the internal or external environment. And then there will be a movement towards the stimulus if it's positive. So a plant growing through life, growing towards light, or movement away from the stimulus if it's negative. So growing down because of gravity. And then there is this video. Let's watch this video to see how plants respond to a stimulus.
If you notice that your sunflowers move a little, you're not losing it. They might just be following the sun across the sky. Storm Team 10's Matt Brooks caught up with a biologist to find out why your sunflowers might be pointing in the opposite direction at the end of each day.
No, you're not going crazy. Young sunflowers really do follow the sun from east to west and back on most days. Biologists and researchers say it has to do with a sort of internal clock.
Just like you and me that regulates our sleep patterns, plants have a circadian clock as well. And in the case of solar tracking, they help to have the plant follow the sun throughout the day.
The sunlight-sensitive leaves, along with the internal circadian clock, help to physically tilt the sunflower.
That causes hormones to be distributed differently on the different sides of the plants, and that causes unequal growth.
You'll find that after a while, your sunflowers may just look east once they're fully grown.
But there's a benefit to them facing east when they're mature, and that's to help attract pollinators in that warm morning sun.
If you would like to learn more about the science of moving sunflowers, just head over to our website, WTHITV.com. In Terre Haute, Matt Brooks, News 10.
Isn't that pretty cool? Most people don't realize that sunflowers will kind of track the sun, and there are several flowers that will do that as well. All right, so reproduction. In order to be a living thing, single-celled organisms or multicellular organisms need to be able to reproduce. Of course, single-cell organisms reproduce by dividing into two. It could be through a process called binary fission, or which is very similar to mitosis, or by budding. But multicellular organisms, they typically produce reproductive cells called gametes. If you remember from introductory biology, those gametes are sperms and eggs, and those will fuse during fertilization and form a new individual or a zygote first. As that organism then grows, it's going to grow and develop over time, and that's called aging, and it is a result of genes providing specific instructions to cells. So as the organism grows, it's going to have cell differentiation, and then that is what will allow it to develop over time. So the cells will increase in size and number, but again, differentiate into specialized tissues in multicellular organisms. Regulation. So organisms require multiple mechanisms to coordinate their internal functions. This could be responding to the stimuli. It could be coping with environmental stress. And a really good example is just consider what happens to you when you exercise. When you're exercising, your muscles need more energy. If your body doesn't increase the blood flow and the nutrient transport to the tissues, then the muscles will begin to rebel, and that's what causes cramps. So it's not, the cramps and the buildup of lactic acid are not the same thing. So if you think about if you've ever done a wall sit or something like that, that is a buildup of lactic acid due to an anaerobic respiration, not cramping. All right, so regulation and homeostasis are typically very confusable for students. So homeostasis is maintaining steady states or conditions within the organism itself. And there are some pretty common things that we typically want to keep at a normal, consistent level. Temperature. Okay, we know that if our temperature gets too low or gets too high, that's a concern. And so your body will actually try and regulate that temperature. Your pH of your body, your blood pH is really, really important and can be affected by how much carbon dioxide is in your blood. How much chemicals you have. So we want to get rid of those waste products and keep a nice balance of chemicals. And then, of course, our oxygen levels we want to keep. So conditions are always fluctuating within the environment, and we'll talk about how we keep this homeostasis balanced. We have receptors that are going to sense when we're out of balance and what will happen and what we need to do to return to balance. All living things process energy, so all organisms use a source of energy for their metabolic activities. Some organisms are going to do photosynthesis and capture energy light from the sun, but some will ingest food and then use just respiration. It is important to know that photosynthesis is just the making of those chemicals, that those organisms that do photosynthesis also undergo respiration as well. We have levels of organization to living things. We talked about these a little bit at the beginning of the slide of the presentation, but also on the first day of school. Living things are highly organized and structured and follow a specific hierarchy that we examine from the atomic to the global in biology. So molecular, we talked about the fact that the atom is the smallest, most fundamental unit of matter. It is not the smallest living thing. An atom consists of a nucleus surrounded by electrons, and atoms bond together to form molecules. So molecule is a chemical structure consisting of at least two atoms held together by one or more chemical bonds. There are tons of molecules that are super, super biologically important. The most important are macromolecules, and those are the big four that we talked about in introductory bio, which are your lipids, your carbohydrates, your fats, and your lipids, which are fats, your carbohydrates, your proteins, and your nucleic acids. So those are your macromolecules and can also be called polymers because they're made of smaller units called monomers. All living things have some type of cellular organization. So some cells contain aggregates of macromolecules that are surrounded by membranes. Those aggregates are called organelles, or tiny organs. Organelles are the small structures that exist within every cell. So all living things are made of cells. The cell is the smallest fundamental unit of structure and function in living organisms. So those are two parts to our cell theory. Some organisms do consist of a single cell, but most are multicellular. Prokaryotes, of course, are single-celled, but they can be what's called a colonial, and we'll look at some organisms later this semester that are colonial, and they are single-celled organisms, but they live in a colony, and they're still prokaryotes because they don't have that membrane-bound nuclei. Okay, eukaryotic cells do have those membrane-bound organelles and a membrane-bound nucleus, but are often multicellular, although not all eukaryotic cells are multicellular, of course. Organisms have organization. So in larger organisms, cells combine to make tissues, and then those tissues combine to make organs, and the organs can combine to make organ systems. And we have talked about that before. And then, of course, our ecological organization, which we've also talked about. All the individuals of species living in areas of population. If you add more populations in the same area, you were talking about a community. Adding the abiotic, non-living parts of the environment now makes it an ecosystem. And then including all the ecosystems is the biosphere. We have a lot of diversity of life. So biology is a broad scope because of so much diversity on the earth. In my travels, even just this past summer to South Africa and to Alaska, just seeing the diversity and the two different areas, and even from the difference between where we are here in Virginia. And evolution, this is the process of gradual change during which new species could arise from older species or even just change on its own. And that's really the source of where we get our biodiversity from. All right, so our advanced objectives in this themes and concepts of biology is really recognizing and interpreting a phylogenetic tree, and then listing examples of different subdisciplines in biology. So talking about our properties, our order, we have cell differentiation. So that's when less specialized cells become more specialized. So you have stem cells, which are less specialized, but they would then become more specialized into either a muscle cell or a blood cell or a nerve cell or so on and so on. And so in muscle cells, in multicellular organisms, this is really important in how we get those specialized cells. How we respond to stimuli. So we have some key words that we typically refer to. And so one is called a taxis. So that's the general response to stimuli, and that's moving in response to a stimuli. And two very common taxis that we look at is phototaxis, moving towards or away from light. So that's what we looked at with the sunflowers. Or chemotaxis, moving in response to chemicals. In reproduction, when that reproduction occurs, DNA contains genes that are passed along to an organism's offsprings, and genetic engineers can actually make or introduce new genes into organisms, and we will become genetic engineers into later in this semester. Viruses themselves can pass along different genes that can be hidden in their DNA from one to the next. So that could be and has been a thought of an aid to evolution and genetic diversity. All cells of all living things use DNA to control their growth and development. So you have controls or switches that are going to tell your body and your cells when to start growing and to stop growing. The sequence of the DNA is different from one species to the next. So you're going to have different controls from one species to the next. And the specific genes and their level of activation determines exactly what the cell will do and ultimately what the organism will make. All right, so regulation or organs are really good at our regulation. That's our key point to regulation. So before I talked about like cramps, but that was part of the blood flow. So our heart as an organ is going to pump blood throughout the body. So that's regulating the blood flow throughout the body. Our kidneys are going to remove metabolic waste. So they're regulating the waste level that's in the body. The blood, again, working with the heart, delivering nutrients to every cell, and the skin cooling the body. So their regulation is very closely tied to homeostasis. One of the key ideas in homeostasis, and a really good example, and I mentioned it before, is your temperature. And the actual term for this is called thermoregulation. And so thermoregulation, being able to control that temperature, is really important. But a major environmental problem or change to an organism is temperature fluctuation. So if you're an organism that lives in a cold climate, you're going to have adaptations that allow you to live in that cold climate. But as temperatures fluctuate and your cold climate becomes less and less cold, this is going to have become a problem for you. You're going to have a problem with thermoregulation. All right, so energy processing, chemosynthesis. So we don't just have photosynthesis and respiration, but we could actually have something called chemosynthesis. And this is the making of organic compounds by bacteria or other living things, not using sunlight. We're using chemicals. This is very commonly done in hydrothermal vents or in the oceanic crust. And then looking at that diversity of life, so a phylogenetic tree, that's a diagram that shows the evolutionary relationships among species based on similarities or differences in genetic or physical traits. So this particular phylogenetic tree here was built looking at similarities in ribosomal structure amongst these different organisms. So each branch is called, has a point at which it branches off of, and that's called a node. And that node represents ancestors. And those nodes are points in evolution from when the ancestor diverged from that new species, or that new species diverged from the ancestor. The length of the branch can actually be proportional to the time since that split as well. So if your phylogenetic tree is very accurate and you're including those branch lengths, it can give you even more information. All right, so back to some other branches of biological study. So molecular biology and biochemistry are going to study biological processes at the molecular and chemical level. That's some of the areas where I get the most passionate about. Microbiology is a study of microorganisms and the structure and function of those single-celled organisms. Neurobiology is studying the biology of the nervous system. Paleontology is using fossils to study the history of life on Earth, or the history of an organism's life. And zoology and botany, zoology being the study of animals and botany being the study of plants. And I think that is all for today. So I hope you enjoyed, took some notes, and if you have any questions, please bring them to class.